Hello friends, welcome to BMH Learning. This lecture is about isopicnic centrifugation, also called as buoyant density centrifugation or equilibrium density gradient centrifugation. In isopicnic centrifugation, the particles are separated solely on the basis of their density. This method of separation is independent of time and relies solely on the actual buoyant density of the particle. Now, what is this buoyant density? The buoyant density of a macromolecule is its effective density in solution. It is defined relative to the fluid medium in which it resides. We can say through which it is sedimented. Now let's get into the details of this method. Let's suppose we have this centrifuge tube. First thing we do is it is filled with a matrix forming substrate through which the particles would be separated. Now, the substrate we are using is actually made of gradient forming substance. But the important and distinct feature of this substance is that we have to choose this matrix forming substance in such a way that it can provide the gradient difference after the centrifugation. So, in case of isopicnic centrifugation, the gradient is not formed in the first place. Now, this is in contrast to the gradients you encounter in sucrose density gradient centrifugation and zonal centrifugation, where we choose the matrix forming substrate in such a manner that it can form gradient in the starting of the centrifugation. The next thing we need here is uniform mixture of sample. The biological sample, for example, here is nucleic acids or proteins. So we put our sample into this column and mix it in such a way that the solute forms a homogeneous mixture with the matrix forming substrate. So the uniform mixture is basic requirement in isopicnic centrifugation. Now, after the preparation of the uniform mixture, we will go to the centrifugation step. So the column containing the mixture is put into the centrifuge. The centrifugation is run for a required period of time according to the sample being used. After the spinning process is done, we are left with this. So what we are getting here? Gradient formation. So the centrifugation process leads us to the gradient which was not present prior to the centrifugation. So now we are ending here with three different bands formed of different particles. Now the thing is, what is the basis of their separation into these distinct bands or layers? You must remember we talked about buoyant density of the particles to be separated. We can understand it this way. The macromolecule is being sedimented through a fluid column in which the density of the column is adjusted. That is, the column has got lowest density at the top and maximum at the bottom, while the middle portion is having intermediate density. The macromolecule moves to a fixed position in the fluid column, or we can say it would move until its buoyant density is the same as the surrounding gradient medium. At this position, the density of the macromolecule is equal to that of the solvent. This is an equilibrium position that is independent of time. By this method, the particle will never sediment to the bottom of the tube, no matter how long the centrifugation time. Now let's talk about the properties of an ideal density gradient medium. It should have sufficient solubility to produce the range of densities required. It must be easily removable from the particles to be separated. The medium should not affect the biological activity of the sample. Also, it should not be toxic and not metabolized by the cells. The medium should be autoclavable. And last but not least, it must be cost effective. This was all. Thanks for watching.